Okay, so today we're going to talk about arterial spin labeling, and then just to sort of give you a flavor, there's way too much to cover in one lecture, and I want to end the lecture with talking about sort of thinking about future directions and some sort of things that maybe it'd be good to think about as you're embarking on your careers in neuroimaging. Okay. So here are the key concepts. So we'll talk about cerebral blood flow, talk about lots of different issues with ASL, and then we'll get into future challenges and directions. So first of all, cerebral blood flow is a fundamental physiological quantity. We all need it, right? Otherwise, we'd be dead. And it's obviously closely related to brain function, and it's basically the delivery of blood and oxygen to your brain tissue. So it's also called perfusion. So you might hear about things like perfusion fMRI. That's basically arterial spin labeling fMRI. And it's actually the rate of delivery of arterial blood to capillary bed and tissue. And these are the units. These are very arcane units, but it is actually from the world of physiology, and it's measured in mLs of blood per 100 grams of tissue per minute. Okay, which is sort of a weird thing, but that's basically how much of your blood are you delivering to some 100 grams of tissue, and how much are you delivering in 1 minute. Okay. So a typical value is 60 mLs per 100 gram per minute, or 60 mLs per 100. And if you assume 1 gram per milliliter, which is a good approximation for brain tissue, then that turns out to be 0.01 inverse seconds. Okay, so that's the rate of delivery. What that means is if I wait 1 second, 1% of, and I look at any volume of gray matter, 1% of that volume is new blood. Okay, so that's sort of how you can think about it. All right. So every second, you're basically replacing 1% of your brain tissue with fresh blood. All right. And if you don't do that, you'll be dead. Okay, or have brain death first, and then. So that's very important. That also sets a limit on why ASL is a very low SNR method, because it means from a volume of brain tissue, only 1% of that signal is going to be fresh blood, and that's what we're trying to measure. Okay, so that's why SNR ASL is a very challenging method, because we're trying to measure that very tiny signal.
How do you deal with like old infarcts and
like dead
tissue?
dead tissue? It's not an easy thing to deal with. So as long as there's being blood delivered there, and that there are and the problem there is that the delivery of blood may take longer than usual, right? So we'll talk a little about collateral flow, sort of down the road. But it is difficult. But as long as something's being delivered, ASL should be able to measure it. Okay, so the traditional way of measuring, or sort of one conceptual way of thinking about it, of how you'd measure blood flows, you could start off, imagine you could inject a lot of little spheres into your carotid, right? Just boom, like that, you know. And then you just measure those spheres going into your brain. And then you might wait some amount of time and you say, okay, on the top, you know, a bunch of spheres arrived, and on the bottom, less spheres arrived. And then, so this, on the top, you'd have more cerebral blood flow. The voxel on the top has more blood flow than the voxel on the bottom. Okay? And that's sort of conceptually what we're going to be doing with our arterial spin labeling. But in the old days, for example, in animals, what they would do is they'd actually inject contrasted, you know, radioactive tracers into the mouse's blood supply and watch it go into their brain, and then it would just get stuck in the brain because the tracers are too big to go through the capillaries, and it gets stuck. And then I love this paper. This is one of the first papers I read when I was getting into the field in 1992. This whole paper is about whether it's better. So basically, you do this, and then you like chop the mouse's brain off, and then the question was, is it better to like strip off the skull and then freeze the brain, and then you can slice it up and count it, you know, with radiography, or is it better to freeze the brain, freeze the whole thing, and then strip things off later? So that was the whole paper's comparing that. So obviously this isn't really a method we can really employ in terms of healthy human subjects, right? We can't be chopping their heads off and freezing their brains. Right? So there are other methods. So that method is called quantitative autoradiography. even if you could do it, you probably wouldn't want to do it because it's such a pain to do it. But there are other methods like laser Doppler. You can measure blood flow in sort of large arteries. You can use radioactive tracer-based methods such as PET and SPECT. You can use contrast-based MRI. But the problem with all these methods is they're very invasive. You're injecting something, and anytime you inject something into the bloodstream, there's always some finite possibility that things are going to go wrong. And also, subjects don't tend to like that, right? So that's why arterial spleen labeling is super important for neuroimaging, because it's totally non-invasive. You just stick the person in, you turn the magnet on, and you get a blood flow measurement. So let's talk a little bit about how it works. And we're not going to give we're just going to give you the broad strokes of how it works. So the basic idea is you're going to look at the spins in the water. The hydrogen spins in your blood, okay, the blood water. And what you do is, remember, way at the beginning of the quarter, we talked about RF pulses and how you could use RF pulses to make spins go in flip them different ways. So we're going to take the spins and we're going to flip them that way. So we're going to invert the spins, okay? And so that's what's called tagging by magnetic inversion, right? And so those spins, if I tag a spin here, it's going to flow up, right? And as it's flowing up, it's gradually relaxing back to where it wants to be. Okay. So then, and what I do is I acquire an image before it's fully relaxed back. Okay. And then I do a second image called the control image, where I don't invert the spin, and that just is fully relaxed. Okay. And so the key behind ASL is the difference between the control and tag images is proportional to how much blood was delivered. Okay. So that's traditionally ASL is always done as a difference between a control and a tag image. Okay. So there's different types of ASL. There's pulsed ASL, which is basically I invert a whole bunch of spins at the same time. And then I let them invert, they flow up and invert, and then I try to acquire my image before they fully relaxed. So this red thing is showing the magnetization of the tag spins, and it's returning back to equilibrium. The control is always at equilibrium. So I have to acquire my image before it's fully relaxed. So that's one of the time constants for ASL, because the blood is relaxing with a time constant of about 1.6 seconds. So for example, if I wait 10 seconds, there's not going to be any signal to measure. Okay. The other kind is something called continuous ASL, which is I tag blood as it's flowing past a plane. Okay. And there's a third kind called velocity selective ASL, which was actually invented here by Dr. Wong at UCSD, which is I tag blood based on how fast it's flowing. Okay, so we're going to go over a little bit of each of these methods because they're all used, they're all available here at the center, and they all have their advantages and disadvantages. Okay. One disadvantage of velocity selective ASL is that currently it's starting off from what's called saturation. It's not starting off at negative, it's starting off at 0 and then relaxing. So the overall signal you can get, that difference, it can be less. Okay, but those of you who were in the previous talk, there are methods to try to address that. Okay, what does a pulse sequence look like in terms of the timing? So typically there's going to be a tag, and then those of you who have used ASL or have been a subject with an ASL know that there's a very particular sound for an ASL sequence, like nothing, like that. You know, there's like a tag or boom, and then there's dead time, and then okay. So that could be an MP range, but it's also very characteristic of an ASL, that there's this dead time. And that dead time is where we're waiting for the blood to flow in. So for pulsed ASL or velocity selective ASL, that tagging happens very quickly, and then there's a dead time, and then we'll acquire, and we'll alternate between tag and control. Okay, typically tag, control, tag, control. For continuous ASL, you'll actually hear the labeling goes on for a long time because we're tagging blood as it's flowing across a plane. So if you listen to one of those sequences, like, you know, and then a long delay, and then the imaging will happen. So that will sound different than a pulsed ASL. Okay, and then you'll do tag, control, tag, control. Okay, so let me just give you a sense of some of what the MR physicists, what we try to do when we're designing these ASL sequences, um, the the idea is, we talked about the tagging, but the control of the tagging is never perfect, and it's the problem that the thing that keeps MR physicists up at night is, does the tag somehow affect my imaging plane, right? I like a pure tag that only tags the blood, but sometimes it also is messing around with the signal in my imaging plane. So therefore, we want to put this far away from the imaging plane, but if we put it too far away, then it takes too long for the blood to get there. So we want to put it fairly close, but then we want to, it still has an effect on these spins, and so we need to, in the control image, we sort of have to balance out those effects. So one way to do it is, you could, in the control, you could just tag above where you care about, because blood generally flows this way, not that way. But that only really works well for one slice. And so there's just these different ways. FAIR is probably the most common one that we use here at the center. So those of you doing pulse ASL at the center will be using FAIR, where the way it does is it inverts everything, no matter where it is. And then for the control, it only inverts in the imaging plane. Okay? And so the imaging, things that are being imaged are inverted both tag and control. So when you subtract that, you shouldn't get anything. But those things that are in the tag those spins are only inverted during the tag. So that's how that works. So that's called FAIR. And then PCOR is another way that uses off-resonance pulses to try to control for that. But there are all ways, the different flavors of pulse ASL are all trying to control for the fact that we can never really perfectly just do the tagging. We sort of somehow have to balance it out with some other method. Okay. Continuous ASL, as I said, the spins flow through, okay, and then as they pass this plane, they get inverted, okay, and so that's the tagging. So one thing you could do is you could just put that plane on the other side of your imaging plane so that the spins don't get inverted, but that only really works for the imaging plane right in the middle. It doesn't really work so well for the other imaging planes. So people over the years have come up with tricky methods. This is one called amplitude modulated control, where it sort of inverts and then reinverts and then reinverts again. So you invert and then you invert again, so the control is not affected. But continuous ASL was one of the first ways ASL was used, but it's never used. Okay, so at least for humans on clinical systems, because the pulses, just the scanners, their RF, the radio frequency amplifiers just can't support it. So you might see these used in animals or with special coils, but for the most part, you'll never see Castle for very standard sequences. Okay, but it was one of the first ways that ASL was done, so it's still talked about. What you will see is something called P-Castle, and that's what you'll see at the center, which is pseudo-continuous ASL. So as I just said, Castle has the problem that there's this long labeling pulse, and so MR systems tend not to like that. If you tell the MR system, oh, just turn on your RF pulse for 4 seconds, it'll just say, no, I'm not going to do that. Okay, and often it just can't do that. So the invention that a group, David Alsop's group, came in is they just took that pulse and they broke it up into little chunks. And so that's pseudo-continuous ASL. And then there's some other tricks they have to play, but that's basically what you'll see on modern scanners today, and is also offered sort of by the vendors now. Okay. So pseudo-continuous ASL or P-Castle. So that's what pseudo-continuous ASL looks like. So we basically break up that RF into little chunks, and that's how that works. Now, velocity selective ASL, which was invented by Dr. Eric Wong here, it's a super cool method which takes advantage of the fact that as blood is going from your arteries to your arterioles to your capillaries, it's decelerating, right? And then as it's going out, it's accelerating, right? So if you can just invert the spins that are moving faster than a certain speed, you're going to mostly invert these spins out here, right? These are the spins that are moving fast, okay, or you're saturating these spins. So that's your tag now. So basically, you are able to, here the tagging cutoff is about 1 centimeter per second. which corresponds to arterioles about 50 to 100 microns. So you're really getting very close to the brain tissue, right? That tag is coming super close, and that's going to be very important when we talk about some of the pros and cons of ASL. So you're not tagging down here and waiting blood to flow centimeters. You're right there, close to the brain parenchyma. Okay. And then in the control, you sort of leave. You just don't do that. And the amazing thing, just the way that, it's amazing that you can get these diffusion tractographies after, 5 or 10 minutes. I think it's also amazing that you can just put someone in a scanner and literally, in as little as 2 or 3 minutes, you can actually have an image of the blood flow in your brain. Okay, so I still find that quite amazing. So this is Castle, PASL, another pulse ASL, and velocity selective ASL. These are like whole brain. That this is sort of, I think, probably acquired with our standard 2D fair whole brain protocol here at the center. And there are lots of really cool variations on ASL. This is just one of them, where also invented by Dr. Wong, where you can actually not only tell how much blood is in a certain voxel, you can figure out which artery it came from. Okay, so in this case, they're color coding left, right, and vertebrals in terms of the different blood, which artery was supplying which territory. So this is what's known as vascular territory imaging. Okay. So one example of that is in this patient here who had stenosis. You can sort of see here that before it, his whole right part, the part that should have been supplied by the right carotid, was probably from the vertebrals, okay? And so the stuff that was supposed to be supplying this part also went and supplied the whole right side. And then after the stent was placed in, then the carotid was able to then supply that territory again. Okay.
I have a question
Yeah.
about that. Is that an additional sequence on the scanner to get those, or is it just the way you process it after you collect your ASL data?
It is it's an additional sequence that's not typically used because there's different variations, but the current variation requires you to select where you think the arteries are. Mm-hmm. So those of you who have done optimized pCASL actually have done this. So that is using, so actually you have that. So if you've done optimized pCASL, you actually have this information. So it's something that actually has, you can, if you want it, it's easy to set up, it's straightforward to set up, and might be useful for certain study populations. But it's just, it's a very cool technique that really has not necessarily found a sort of, the clinicians haven't really adopted it yet. So it's one of these things which seems really cool. Everyone loves it when they see the pictures, but has not really been used that wide clinically yet. And potentially because of workflow issues. Okay, so as we talked about before, the ASL signal, the delta M, is really the difference between the control and the tag. And so the delta M is what we measure. What we want to estimate is cerebral blood flow. And so there's a lot of sort of math and physics that goes into figuring out what's the proportionality factor to get you from your delta M to your CBF. Okay, so I'm not really going to go into that except to say that there's a lot of factors that lots of people have thought about over the past years, and it depends on both physiology and MR physics. And so it's one of these things where the people figuring this out have to know enough about the physiology and the physics to sort of put those together. But if you do everything right, then, and the whole idea is to make that quantity fairly robust to variations in physiology, because you don't want that parameter to vary between old and young, or low and high CBF, right? You want that to be fairly robust to physiology, because otherwise you're not going to be able to measure physiology correctly. So a lot of the work over the last 20 years has gone into how do we make sequences where that A effective is robust. Okay. So there's a lot of sources of errors. There's transit delays, bullets with relaxation effects, intravascular signals, offsets, off-resonance effects, velocity sensitivity. So these are all things that sort of MR physicists worry about. I'm just going to talk about sort of the main one, which is really transit delays, because that's something that can vary a lot with physiology, and can vary a lot with patient groups, and so something that we need to think about. So here's the basic idea: is as we talked about for Castle or P-Castle over PASL, our tag, for reasons because we don't want to have too much interaction between the tagging and the imaging plane, there's got to be some distance between the tagging and the imaging. Otherwise, things just don't work. Okay? And the problem is now it takes time for blood to flow that distance. And that's what's known as the transit delay. And what's more, it takes more time for blood to flow from here up to the top of the brain than it does to the bottom of the brain. So even across slices, you're going to have issues of these transit delays. Okay? So I'm just going to, I realize this is a fairly complicated slide, but let's just look at the top one. So here it is for Castle or P-Castle. So we tag the blood here or label it here, and then this PLD stands for post-labeling delay. That's the amount of time we're waiting for the blood to go in. And the key is we want to make that time long enough such that we know all the blood has gone there. If we don't wait long enough, some of the blood might still be in transit, and we're not going to measure it. Okay, so we're going to underestimate CBF. All right? And so if you look here, this is from what's called the white paper. So there was a white paper written in 2025 saying, what should be the standard parameters for people to use clinically. And you'll see here, these are the PLDs that they're specifying. Okay, you notice here that for children, it's relatively short, 1,500 milliseconds. Okay, they tend to have smaller necks and heads, and also their blood flow is pretty fast, and so you can get away with a smaller PLD. In typical adults less than 70 years, it's recommending 1,800 milliseconds. So it's saying, you're tagging your blood and then wait for 1.8 seconds before you acquire your image. Okay? You'll notice that for adults greater than 70, they're recommending 2,000, and also for clinical patients, 2,000. Okay? And that's because older subjects tend to have lower blood flow and longer transit delays. Okay? So if you use the 1,800, you're going to underestimate blood flow in these subjects. And for example, if you're comparing, healthies versus older subjects and use 1,800, you would, just because of that error, you would measure lower blood flow in the elderly adults, and you would be overestimating how low it is. Okay, so it is important to make that delay long enough. For pulsed ASL, this is sort of the equivalent to the PLD. Okay, because this is sort of your tagging time, and so in this case, basically, this TI says use the PASL PLD for that. So because in this case, yeah, so actually that's a mistake. Let me correct that. Whoops. Okay, well, let's just cross that out because I can't get the eraser. So the inversion time, this is the PLD here. Okay. So once again, we have to wait long enough for the blood, because there the blood is all tagged at one time, and so we have to wait for it to get into there. Okay, so if you look at what's available at the center, these are the various versions available at the center, and so you'll see like the standard with a PLD of 1,800, pediatric 1,500, geriatrics 2,000. Okay, so these are essentially the white paper. So if you pick these protocols, you're essentially picking what the white paper said to use. What else? You'll notice we do also have something called the white matter protocol, and that you'll see has a fairly long post-labeling delay as well. In addition, it uses what's called background suppression pulse. Okay, and the idea is that blood flow in white matter is even less than blood flow in gray matter, and so you have even less signal. And so we use something to sort of suppress the signal from the tissue, because that's going to subtract that anyways. But if it's a big signal, so instead of making the signal just 1% of the overall brain tissue, if we can reduce the brain tissue signal, then we can make it like 10% or 20%. And so that can help improve the sensitivity. So that's one protocol that some people might be using. Yeah.
For white white matter, would 1900 work for geriatrics as well, or would you have to increase that even further?
Oh, you mean for white matter and geriatrics?
Mm-hmm.
Yeah, you'd probably want to increase that even further. Let's see what I did with my water. Well, I'll find it when you guys are doing your quiz. Okay. This is an example of what happens when the PLD is too short. So you'll notice. So this is on an 85-year-old man with a labeling time of 1,500 milliseconds and a PLD of 1,500 milliseconds. So there, you'll notice the blood is still in the arteries, okay, because it hasn't had enough time to go through the arteries to the tissue. So if you did the measurement at this point in time, wherever that blood was going to go, you would measure, your CBF measurement would be too low, right? Because the signal that was supposed to get there didn't get there. Okay, so that's why transit delays are sort of this issue that people keep worrying about in terms of, what should you do? And the problem is that there's really not a great answer right now. There are other methods that we won't talk about where you could do multiple inversion times, but even there are issues. So we'll talk a little bit about what to do to address that. It's even more important clinically in certain populations. So for example, I believe this is in a subject with moyamoya disease. So this is a paper from Dr. Bolar, who's on our faculty here. So in a normal subject, you label the blood, and it all gets to where you want it to go. And so you're good, and you have a good CBF image. Okay. But let's say there's a stenosis here, and so the blood has to go through this collateral flow. Okay. So now, and some of it still might go through the normal route, but now most of it's going to take much longer to get through all that. And so when you measure it, a lot of the blood is still on its way, right? It's either gone through this collateral flow, or it's just moving very slowly because after it goes through stenosis, its velocity is slowed down, right? And there's less blood flowing there. So you'll notice here there's these big deficits where it's not that the blood flow is lower in those regions, it's just stuck in the supplying vessels. Okay. So I believe this is an animation that Dr. Bowler made. So that's just basically it. Just didn't make it there. Okay. And the problem is we can't wait too long for the blood to get there because the signal is dying away with the T1 of blood. So it's dying away with a time constant of 1.6 seconds. So if it takes the blood 8 seconds to get there, we have no chance of ever seeing it because our signal will have died away with conventional ASL. Okay. So the hope is, and the work that's currently being done here at UCSC and other places, is to make velocity-selective ASL the next generation. So it may be that the next time the white paper recommendation comes out, the guess is it will probably be more velocity-selective ASL than pCASL. Okay? And the idea here is, as we talked about, we're tagging very close to the brain tissue. We're at the 100 micron diameter arterioles, so we don't have to worry about this transit delay. We're tagging blood wherever it is, wherever it's moving slowly. And so that allows almost immediate delivery, and so in theory, gets around these transit delays. Okay. So this is an example in a moyamoya patient, and these are, as you can see, these are angiograms showing blood flowing in. So you see at 1 second, a lot of the blood is making a good, is doing well here, but there's obviously impaired delivery on this side. Okay, and you can sort of see here in the CBF maps done with conventional ASL, there's these big deficits in blood flow because the blood is still stuck in the arteries. Okay, down here, same subject, it looks better, right? So this is from a clinical point of view, this is really important because the diagnosis of someone with no blood flow versus just delayed blood flow is actually going to have different consequences. Okay, so I'm going to go pose you a question so I can go drink some water. And let me just make that quiz available. So this is, what quiz is this? Oh, ASL methods. Okay, great. Can people see that now? It should be called ASL methods. Yeah, everyone see. Okay, so go ahead and feel free to talk to your neighbors. I'm just gonna grab my water. Without? Whoa. That would have been fun. Yes, thank you. Okay, let's take a look. It looks like most people have responded. So, um, so great. I think most people there's pretty good agreement. So, but let's just go through it. So, first of all, what does PCASL stand for? Right, pseudocontinuous ASL. So that's where we use a train of RF pulses, right? So that would go here. So, and it has higher SNR, but it is sensitive to off-resonance effects and velocity. So one issue is, for example, if you have someone with a lot of dental work, that can actually affect the tagging because that tends to be close to the carotids as well. So that's something where if you know you have a population with a lot of dental work, you would probably need to use what we call, at the center we invented something called optimized PCASL, which actually tries to correct for that. Okay, and also the other thing is we'll talk a little bit later about doing functional MRI with ASL. A lot of scans will just maybe do 5 or 10 minutes of ASL, in which case you don't necessarily want to go to the trouble of correcting for all these issues. But if your whole study is based on ASL, let's say you're going to do an hour of ASL, then it becomes worth it to spend 5 minutes making sure you're really correcting for these issues before you spend the next 60 minutes correcting bad data. Okay, so there are trade-offs in terms of even ASL. Okay, VSASL, what does that stand for? Velocity selected, right? So therefore, there's only one has velocity in the name, so that must be that. This is really great, and I think this is probably going to become, hopefully, if we can solve all the problems, will be the go-to clinical and potentially research application. Currently has lower sensitivity, and there's also issues where it's very sensitive, because it's based on velocity, it's very sensitive to anything that's moving in the brain, including CSF pulsations, okay? And therefore, actually, in older populations, it actually really has a problem because you got a lot of CSF in these older folks, right? So that's currently what people at the center are trying to figure out how to deal with that. CASL, what's that stand for? Continuous ASL. So that's a long duration RF pulse. As I said, hard to implement on clinical systems. You can never ever really see it except for very boutique systems. Okay, and the last one is PASL, which stands for pulsed ASL, right? So here, short duration pulse to invert the blood. It's got lower SNR, although the talk that was just given shows that there might be ways of improving SNR. The great thing about PASL, it's fairly robust. It's just really hard to break it. And so in general, if you're starting off a study and you're seeing weird effects with some of these other methods, you might want to always do a PASL sequence. So whenever we're designing something new, we'll generally do a PASL sequence just to get a baseline of, okay, this is what, because we know it always works, okay. It may not be the best, but it's sort of like, you know, it's very reliable. Okay. All right, any questions before we move on? Okay. So I'm going to spend some time talking about functional ASL. So as we talked about, we typically do tag control, tag control, tag control. And so you'll get this raw ASL time series where all the greens are the controls and all the blues are the tag. And this is for a single voxel now. It turns out, and as we said, if we do the control minus tag is your difference signal. So you can just do a running subtraction, right? And if you do that, and there's different ways of doing that, but you can get a perfusion time series. Okay, so you actually get a time series of what cerebral blood flow is doing over time. Okay, so this is what it looks like. So this is from a voxel here in visual cortex. This is the ASL time series. Okay. Now the really cool thing is if I do a running average, I get bold. Okay, and this is the subtraction, and I get CBF. So, and you can sort of see, you know, the bold. One thing you'll notice is the bold obviously has much better SNR, right? It's just up and down, which is why everyone loves bold, right? And the CBF is much noisier, but you can still see the blood flow is going up and down, up and down, up and down. Okay, so this is one of the amazing things you can do with these sequences. Is from the same sequence, you can actually get bold and a CBF in the same sequence. One of the advantages of ASL over bold is that if you're doing anything where things change very slowly, okay, then ASL actually turns out to be better than bold. Because as we talked about, bold has these low frequency drifts, right? So like if you were to do a bold scan, like if your task in bold was like 30 minutes on, 30 minutes off, no one would believe that you could do that, okay? But you can do that with ASL because you're actually measuring blood flow. So you can compare blood flow today and tomorrow. So for example, this 24-hour task is comparing, you know, tapping your fingers today, resting, you know, for the next 5 minutes, and then tapping your fingers tomorrow. And so you could do a block design over days, okay? Because it is a physiological phenomenon you're measuring, whereas your sensitivity bold is basically you have no sensitivity with bold because you know you can't come in and have someone tap their fingers today and then tomorrow have them and just rest and think that that subtraction has any meaning in bold because bold is very arbitrary. It's a percent change. Okay, so that is one of the big advantages of ASL. So anytime you're doing something that has very long time scales, so for example, one thing that are things like meditation and pain. Okay, so these are things that tend to have very long time scales. And so this is studied by Dr. Zaidan, who's now at UCSD. This was done when he was at Wake Forest, using ASL to look at sort of response to pain under different conditions, meditation, placebo, sham control. Okay, and there he's taking advantage of the fact that, you know, ASL is inherently more robust to these very low frequency confounds. And so if you are doing a study that has these very long time scales, it's ASL definitely something to consider. The other area that has received a lot of attention, especially here at the center, is the whole idea that, as we talked about in the second week, bold fMRI is not quantitative. Okay, bold is here, and here's neural activity, and it depends on all these things in the middle. Okay, and typically all we measure is bold, and then we try to make inferences about neural activity. So the idea is, well, maybe you can't measure everything, but if you can at least measure one more thing, then maybe you can start making inferences about, say, metabolism, and that's slightly more tied to neural activity. And both blood flow and metabolism are more tightly tied to neural activity than the bold signal is. Okay, so that's the whole idea behind CBF as trying to get more quantitative. So we talked about before, the bold signal depends on the balance between CBF going up and CMRO2 going up. Okay, one drives deoxy down, the other drives deoxy up. Okay. This is a nice example here. So let's say you compare a young adult versus an older adult. Let's say they have the same neural activity. Okay, but in the young adults, the vessels dilate very well, the blood flow responds very well, and so they get a nice bold signal here. Okay. In the older adults, you might have impaired blood flow because their vessels are stiffer. Okay. They might actually also use more oxygen metabolism, and so they are going to have a smaller bold signal. Okay. So even if their actual neuroactivity was the same, their bold signals could be quite different, and it'd be very hard for you to interpret, although you can always interpret anything, but your interpretation might be wrong. Okay. Yes.
Can you use CBF to regress out your fMRI data?
Yeah. So one thing you can do is if you don't have the time or the sensitivity to do the bold and CBF at the same time, then I think what most studies should do, although even large studies aren't doing it, like I don't think ABCD is acquiring any ASL, is it? Yeah, which I think is ridiculous. Sorry.
I'll let them know.
Okay, well they probably don't. Because blood flow obviously is changing with development, and I think it is because it's going to change your baseline deoxyhemoglobin levels, it's going to change a lot of things. And so I think if you don't know what baseline blood flow is doing, then you are really missing out on a huge physiological change that is going to affect your bold signal. Okay. So here's an example. So this is what we did. This is a study that Cal Rustum and Katie Mangan did back in 2007. So we looked at CBF in the hippocampus in young and old subjects, and as expected, the older subjects had considerably less blood flow than the younger subjects. When we looked at their bold signals, they were slightly different. Maybe the older subject had a slightly greater bold signal, but it wasn't statistically significant. But when we look at their percent change in CBF, we saw a much bigger change in the CBF response of the older subjects. Okay, and so when we went through the modeling, we basically said that it looked like the percent change in metabolism was much greater than the percent in the older subjects than the younger subjects. Okay, that basically the older subjects were having to work harder, and it turned out that it seemed as if with some modeling, if the older subjects were starting off at some baseline metabolism and the younger were here, they were both trying to get to the same level, and that's why the older subjects had to work harder. So when we looked at where they were trying to go, it looked like they were trying to go to the same place. Okay, and so this is a interpretation that would be very hard to get from bold alone, right? Because you don't have enough information, right? From this, you might even say there's nothing to report. Okay, but knowing that there's So first thing is just knowing that there's a change in baseline CBF might lead you to think, well, maybe that's not the whole picture. But then if you can also measure this in addition, you get a much richer interpretation. Okay, so that's something that is something that I think is good to keep in the back of your minds as you are interpreting your bold-only studies, that there might be something else going on. As we talked about before, I mean, one of the disadvantages with ASL fMRI is the SNR is really bad compared to bold, and also in general, it's harder to do whole brain coverage, and small voxels and things like that. So that's still why bold is much more heavily used. But that's why things like noise reduction are really important. So for example, we can go from a noisy voxel like this to a cleaner voxel like that. Okay. The other thing that's really kept sort of these methods from emerging or being more widely used is because to really do it right, you have to have people breathe carbon dioxide as sort of a calibration method. So this is for calibrated fMRI. Okay. So the current methods to really get everything calibrated and really measure, and this is to get, from bold and ASL, you can measure CMRO2. You really need to breathe these gases that sort of change your metabolism in a certain way. But people are working on including faculty here at UCSC gas-free method. And so if these work out, then potentially down the line there will be ways of doing this totally non-invasively, in which case they'll be much more attractive to studies. So I would say this is fairly limited methods now, but I think it's just good to keep in mind as you're progressing with your career. Okay, any questions? Okay, so now we're just going to spend a few minutes talking about what sorts of things to think about for the future. This is a blog I took from Peter Bannattini's Brain blog. So this is basically a chapter he wrote that's in his latest book on fMRI, 26 Controversies and Challenges in fMRI. So you can read this online. Obviously, we don't have time to go through all 26 challenges and controversies in fMRI. I'm only showing you this slide to show that for those of us who think a lot about these things, there's a lot of problems with fMRI. Okay, and it's good to be aware of some of these. So I'm just going to go through a couple of them just to give you a sense of what sort of those of us who worry about these things think about. Okay. One thing that our lab is really focused on, especially for resting state fMRI, is the fact that within a 5 or 10 minute scan, the arousal or the vigilance of the subject changes quite a bit. And even between subject populations, there are very different, can be very significant changes in vigilance. So what we found is that, and this is using taking EEG and fMRI and doing EEG fMRI. So basically measuring EEG within the magnet, and that is very, very challenging and time-consuming, but for this study we felt we had to do it because EEG is still sort of the gold standard for how do you assess vigilance and sleep stage and things like that. So what we found is that as subjects become less vigilant, so say low vigilance, then they have much more global activity and the functional cognitivity is higher. As they become as they become higher vigilance levels, then there's much more of these anti-correlations, much less global activity. So a lot of the differences that people are finding in connectivity between populations or across conditions, we think there's a huge contribution of vigilance things. And one way we went about this is one way to easily go from here to here is just give people caffeine. Okay, that's how we actually got into this business. We gave people caffeine and we realized it had this huge effect. And then when we went to the EEG, we could sort of see the effects of caffeine on the EEG as well. Okay, so I think that's one thing, especially those of you who are doing primarily resting state, be aware that there is this emerging area really looking at arousal effects. And even more that it seems like a lot of the, quote, motion-related artifacts that people have been publishing, especially like Jonathan Power, there is just a recent paper coming out, it's already on bioRxiv, showing how a lot of those effects may just be due to arousal effects. Okay, they're not really motion effects, they're really arousal effects. Okay, so I think that's especially important for populations where let's say, in development, there's going to be changes in vigilance, and then especially then on the other end, as people get older, there's going to be changes as well. Okay. This is just showing that even during the course of a scan, the blue is showing vigilance going up and down, and the red is showing the average bold signal going up and down with the vigilance. Okay, and one has been inverted, so they're actually anti-correlated. But there are these patterns where the brain goes into patterns of high vigilance and low vigilance, even within the course of a scan. Okay, so a lot of the resting state fMRI activity that most of us are measuring are very driven by these moment-to-moment changes in vigilance that are just being appreciated. This is a paper that should be coming out in a couple of weeks that where we review all those areas. And so for those of you who want to know more about that, that's summarized in that article. One good thing we can do, though, is you can try to, from the fMRI data alone, the question is, can you predict vigilance changes? And so there are approaches. So, for example, here in blue is the actual measured vigilance changes going up and down. And the red is, if you just use the fMRI data alone to predict what those vigilance changes are, you can, in certain conditions, actually predict fairly well. And so this might be something that, as this method becomes more developed, you could apply it to some of your studies and sort of try to estimate, okay, was this subject really going up and down in their vigilance, and was that what was really driving my fMRI result, as opposed to some other effect? Okay. I think the other area is laminar fMRI, and so this really requires high field fMRI. Okay. And so right now, for example, at the center we have 3T scanners. I do think that for the next 5 or 10 years, there are already 7T scanners available clinically now. There's a number of sites that have. There's probably about 100 in the world, 7T scanners. And so I think that's one area that we're going to have to start paying more attention to. And what this allows us to do is allows you to sort of get your voxel size small enough that you can actually start differentiating activation in different layers of cortex. Okay? And that's really important for modeling because there's feed forward and feedback connections, and there's a lot known about how those are related to the layers. And so if you don't have that resolution, you're never going to be able to answer some of the deeper questions. Okay. This is just showing some mapping of auditory cortex where they're basically able to measure from superficial to deep cortex and do sort of look at how different layers of the cortex have different roles for auditory mapping. This is another study where they were trying to test out a theory of, how is information integrated across episodes. Like, for example, let's say you see someone. One day you see someone a man walking out of a house and getting into a blue car, right? And the next day you see a woman walking out of the house and getting into that same blue car. So those are two pieces of information, and somehow your brain integrates it. Oh, I bet the man and woman, maybe from the same couple or something like that. Okay. Or let's say the woman didn't come out of the house; she just went into a blue car at some other place, at the shopping store or something like that. So we're able to integrate that information very well, right? And the question is, how does the brain actually do that? And so this was a study trying to relate it to different layers of entorhinal cortex and using 7T fMRI to sort of parse apart these different hypotheses of how the brain might actually do that. So this is a study that would be very difficult to do at 3T because you wouldn't really have that resolution. Okay. The final thing I'm going to talk about is more sort of generic and sort of, we alluded to it a little bit, but it really is this whole issue of, how should we be responsible scientists and how should we do our research? And, even in this course of just these 10 weeks, we've exposed you to so many methods out there, right? I mean, there's just way too many methods, way too many analysis packages. I mean, there's way too much for any one person to really know in neuroimaging. And so, for example, this was a nice study by Russ Poldrak. He's been one of the leaders in this area, showing that if you just looked at, I think this was FSL or something, but if you just look at all the different knobs you can pick in FSL, there are like 68,000, or what is it, 69,000 ways you can analyze your data, right? Which is a lot. Okay. And, I'm sort of, I have never done this study, but my guess is, when you see a paper that's published in a journal, I bet they've at least analyzed it at least 20 different ways. Probably, until they got the result they wanted. Okay. This is just another example from another paper showing all the different ways of regressors you can pick for cleaning up your data. Okay, and this isn't even that many, but there's still lots of different ways. Okay. This is another study showing that if you used AFNI, FSL, or SPM, you would get, at least for this subject, quite different results. Okay. So I think there's this whole issue of, science in general, there's this concern about replicability and reproducibility, not just in neuroimaging, but psychology, the behavioral sciences, um, and so I think it's something that is continuing to be an issue. I think there are things that people are trying to address this. So there's things like the Open Neuro, there's the BIDS format, and so I think we are sort of getting there. But I think there are still a lot of sort of environmental, political factors that push people to do their own thing. Okay. I think the other thing is, this is showing looking at, as a function of number of subjects and number of tests, the sample size needed for 80% power. So this is how many subjects you need. And then the idea is, obviously, as you get more and more subjects, right, things like ACP and ABCD and UK Biobank are getting, 1,000, 10,000, 100,000 subjects, right? So orders of magnitudes of number of subjects. Those are very powerful, right? And they're driven a lot by, you want to do genetic analyses and things like that. But what we're going to find is we're going to have results that are very small effect sizes, right? And there's going to be more and more of these as these large studies happen. And so the question is, what does that mean, and how do we do with that? And is it actually telling us anything that is meaningful? So that's another thing in terms of reproducibility. So I think that the other thing is obviously, there is a strong bias to publish only positive results, right? And so a lot of what we know about the brain is only positive results. And so that's something to keep in mind, even in your own work. Obviously, I have done this. Some of you might have done this, the p-hacking, right? So, you can sort of pick, work your analysis until you get the p-value you want or the effect size you want. So I do think that all of us have to be aware of that in our own work and as we're evaluating other people's work, there is the multiple comparisons problem that everyone knows about, which is, we try to correct for it with, alpha-sim, or we do permutation tests. I think the real multiple comparisons problem is the fact that people analyze their data many different ways before they publish, and that's really not reported. So I think that's just something. I'm not sure what the solution is for that, but we should be aware of that. And this is the last slide for the course. I think I already showed this in the first or second week. I think, so what we try to do here is at least give you some of the basics that you could try to then at least understand the general frameworks or the general concepts behind a lot of the software you're using. As I said, there's no way any one person is going to be able to know everything. I mean, there's millions of lines of code in these software programs. So I do think we're going to have to help each other, share the expertise. But each person, especially as you become PIs, it can be a little tricky. You're going to have to stay on top of the field enough to know, what things are being done. It's going to get even worse with deep learning, right? Your students are going to come to you with deep learning results, and you'd be like, I don't know, So I think it's going to get more challenging, and so the more we can arm ourselves with sort of basic principles, and I think that's sort of only going to be helpful. Okay, so are there any last